So I'd just like to start by thanking you all for the invitation to come and talk at this seminar series, which is a really fantastic initiative. As Lidem mentioned, I'm a clinician, so I'm a neurologist, but I'm also an academic. So I run a laboratory at the Institute of Neurology, which is part of UCL, as well as being an honorary consultant neurologist at the National Hospital for Neurology and Neurosurgery, where we deliver a national service for people with mitochondrial diseases. And that's really primarily looking after adult patients, because I'm an adult neurologist. And that really forms the basis of much of the research that I do, including the work that I'm going to present to you today. So I thought I'd just provide an overview of the Institute of Neurology and the National Hospital for Neurology and Neurosurgery, just to give some background to this particular project. For those of you who are not that familiar with mitochondrial diseases, I thought I'd just give an overview of the genetics and phenotypes that we see in adults predominantly, but also children. Then I'm going to talk about the paper that we published in Annals of Neurology, which evaluated mitochondrial DNA variants in exome sequencing data stored at the UCL Queen Square Genomics Facility cohort, which includes a spread of neurological diseases, not limited to mitochondrial diseases. And then I thought, just to whet the appetite, I'd talk about a new study that we're involved with, that we're just in the process of drafting the manuscript for. I realized that when I was preparing the talk, that if I was to talk about both, it would probably last about an hour and a half. So rather than put you through that, I thought I'd just introduce the topic, and if it sounds interesting, perhaps when we've published it, I would be happy to come back and present that work in more detail. So this is Queen Square in central London. The hospital is the red brick building, the attractive Victorian building in the foreground, and then you've got the Institute of Neurology next door, which is a less attractive, brutalist looking building, which is where all the wet labs for the research are. And at the hospital, it's a purely neurological hospital, so it's a very much a specialist quaternary referral centre. And the Institute of Neurology is focused just on neurosciences research. And it's not just the proximity of these buildings that's important, it's also their ethos, which is very much around integrating clinical work and research in neurological diseases for the benefit of patients. So the Institute of Neurology was first established in 1950 and later in the 90s merged with the university, and it's comprised of a number of different academic departments which really spread the whole gamut of different neurological diseases, and they've really formed the basis of the cohort that we analysed for this particular study, particularly those PIs that are interested in exome and genome sequencing projects. But really the mission of the Institute is to translate neuroscience discovery research into treatments for patients with neurological diseases. So everything we do is very much geared towards that, right from discovery science all the way to clinical trials. And in terms of mitochondrial disease research at Queen Square, this was really pioneered by these two people, so Professor Anita Harding and Dr. John Morgan Hughes, who not only identified the first mitochondrial DNA mutation linked to human disease, but also characterized a number of different mitochondrial diseases, both at the genetic and the clinical level, and in addition to a number of other genetic muscle diseases and neurogenetic diseases. And we've tried to continue on this tradition in mitochondrial disease research. And I've been quite lucky really to be involved in all aspects of this. But the one I'm going to be talking to you today is very much focused on genomics and gene discovery. So as I'm sure you're all aware, primary mitochondrial diseases are genetic diseases which affect oxidative phosphorylation. So the ability of the mitochondria to produce energy in the form of ATP by aerobic respiration. And they're really interesting diseases because they're caused by mutations in two separate genomes, in both the nuclear DNA, which is 99.9% of your DNA, but also mitochondrial DNA, which is a very small amount of DNA, the other 0.1%, which resides within the mitochondria themselves. And you can have multiple copies of this very small circular genome, which only comprises 16,500 base pairs encoding 37 genes in each mitochondria. And you can have hundreds of thousands of mitochondria in each cell, depending on its energy requirement. And so because of this situation of ploidy, so you've got multiple copies of the mitochondrial genome, you can end up with a population that's mutated and a population that's not, and that's known as heteroplasmy, which I'll come back to. The adult prevalence is around 1 in 4,000, which sounds relatively common, but actually that's as a group of diseases, and we're talking about hundreds of different diseases. So actually each individual disease caused by individual genes is ultra rare. And as many of you are aware, what's very characteristic of mitochondrial diseases is their very varied clinical presentation. They can essentially affect any organ in the body and with onset any time in an individual's lifetime. So the first mitochondrial DNA mutation, given its DNA, it can mutate, was identified in 1988 at the Institute of Neurology by Ian Holt, Anita Harding, and John Morgan Hughes, and was published in Nature, and was identified in a group of patients, adult patients, with a syndrome known as chronic progressive external ophthalmoplegia, which is a slowly progressive reduction in eye movements and drooping of the eyelids, or ptosis. And what they did is they extracted the mitochondrial DNA from the muscle tissue, and that's important. It was the muscle tissue rather than the blood of this group of patients. And they applied a technique known as multiple restriction endonuclease analysis, which is essentially a method where enzymes cut the circular mitochondrial DNA and effectively linearize it and run it on a gel. And this is taken directly from that paper from Nature. And what you can see are the patients and controls run on a gel. This is a southern blot. And you can see in the patients, there's a second band. So the higher band is the wild type mitochondrial DNA, which has been linearized and run on the gel. And then the second band is run further on the gel, indicating that it's shorter, therefore moving faster. And that's because there's a large amount of that DNA has been deleted. And so you've got a smaller mitochondrial DNA molecule, which is shorter when you linearize it, and it runs faster on the gel. And you can see in the controls, the healthy patients, there was just the one wild type band. What's interesting is you can also see in these patients another phenomenon that's important with mitochondrial DNA, which I've touched on slightly, and that's the fact that they're heteroplasmic. You've got these two clonal populations of DNA molecule within the same cell. But also the bands are running into different patients at slightly different levels, which means some of the deletions are bigger and smaller than others. And also some of the bands are denser, they're thicker than the others, again, indicating that the level of the mutated mitochondrial DNA molecule in those patients is higher, which is why the band is darker and thicker. And we now know that the size of these deletions and the level of these deletions are important, and they correlate with the progression of this syndrome during an individual's lifetime. And so since that first mitochondrial DNA mutation was described, a whole host of different other mitochondrial DNA mutations have been reported, despite this being a very small DNA molecule. And the reason for that is the high mutational rate of mitochondrial DNA, which is related to the fact that it's not protected by histones like nuclear DNA. It's sat right next to a series of enzymes that are producing reactive oxygen species, which are potentially damaging to the mitochondrial DNA molecule. And it also has quite poor repair mechanisms. So when it does become mutated, it's not able to repair itself. And we number these mutations in an anti-clockwise fashion around the circle. So you often hear about different mitochondrial DNA mutations with a number. And the most common would be the 3243 mutation, which occurs in a tRNA gene. So mitochondrial DNA codes 24 tRNA genes, 22 of which are tRNA genes, and this is one of the tRNA genes for leucine, and a mutation occurs there at position 3243, which causes a syndrome called MELAS, which I'm sure many of you have heard of, so mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes. And as I'm sure you all know, mitochondria are maternally inherited, so if a mother has a mitochondrial DNA mutation, she'll transmit that to all her offspring. What you can't predict is the level of the mutations that she'll transmit. So some of these people will have a high level of mutation, therefore probably have a severe clinical phenotype. Others have a lower level of mutation, and they may have very minimal or no problems whatsoever. And that's why you see, even with the same mutation within the same family, often a very variable clinical manifestation. Another important property of mitochondrial DNA, on top of that heteroplasmy, is a biochemical threshold that occurs within different cells. So a certain threshold of mutated mitochondrial DNA molecules has to be breached before you develop clinical manifestations, and that level can vary according to the tissue type and the mutation. So just to recap, mitochondrial proteins are under dual genomic expression. So you have hundreds to thousands of mitochondria in each cell in the body. Depending on its energy requirement, each mitochondrion has multiple copies of a circular genetic molecule called mitochondrial DNA, which encode 13 subunits important for oxidative phosphorylation and the translational machinery required to synthesize those subunits. But nuclear DNA is also very important. In fact, 1,500 or more nuclear genes encode mitochondrial proteins, and over 350 of those have been linked to different mitochondrial diseases, and they're required for the maintenance of that mitochondrial DNA, so its replication, for the assembly of the multimeric subunits of the respiratory chain, and also a large number of the subunits themselves. So mutations in both of those genomes will cause mitochondrial diseases. And so, as you might expect, mitochondrial diseases can present in a whole host of different ways because mitochondria are ubiquitous. But actually, what we tend to see in adult patients is quite tissue-specific phenotypes, particularly brain, muscle, peripheral nerve, and heart, so high energy requiring organs. And what we see is a transition from the more severe multi-system phenotypes in children to the more tissue-specific phenotypes in adults, particularly affecting skeletal muscle, but often the central nervous system at all. So there is a genotype-phenotype correlation between those that occurs with some genes. And one important property is the fact that children tend to have mutations in nuclear DNA, so the majority of children will have a nuclear gene that is causing their mitochondrial disease, whereas the reverse is true for adults, so the majority of adults have a mutation in the mitochondrial DNA. So the age of onset of the disease is important in terms of stratifying your genetic testing to one genome or another. And in terms of the adult presentations that we see, neuromuscular presentations are common, whereas stroke-like episodes are less common, but certainly are very severe and can be life-threatening. So traditionally, the investigation of mitochondrial diseases has been a fairly complex process, and often occurs two things happening in parallel. Firstly, looking for mutations in blood, particularly the most common mitochondrial DNA mutations, and the common nuclear genes that cause mitochondrial diseases. And coming back to that important caveat about mitochondrial DNA in blood, so as you, because of the high replicative rate of blood, there's a negative selection pressure against mitochondrial DNA mutations in blood. So often you don't see novel or rare mitochondrial DNA mutations in blood in adults. And you have to look in a post-mitotic tissue like muscle. And so in parallel to looking at the blood, we often take a muscle biopsy and look for these mitochondrial DNA mutations that may not be detectable in blood, which isn't all of them, but many of them. And we look at that in muscle as well. But muscle is also very useful for performing histochemical studies for different mitochondrial enzymes, performing spectrophotometric analysis of enzymes involved in oxidative phosphorylation to measure their activity, and other studies like Blue Native Gel, and as I say, rerunning a full mitochondrial DNA sequencing just to be absolutely sure there isn't a mitochondrial DNA mutation that was undetectable in white blood cells. As with all genetic diseases, we very much moved towards a first-line genomics approach for mitochondrial diseases, and in the UK, that involves now clinical clinical whole genome sequencing as part of the 100,000 Genomes Project, which I'm sure many of you are familiar with. And as I'm sure you know, when you perform whole genome sequencing, actually you sequence both the nuclear and the mitochondrial genome, even though the mitochondrial genome isn't enriched during whole genome sequencing. There's just so much of it. You will sequence the mitochondrial genome, and often to a very high depth, up to 2000x, when you're doing it using whole genome sequencing. And so, as part of this project, we decided to look at the mitochondrial DNA from exome sequencing data, so not genome sequencing data, but exome sequencing data in a large number of neurological diseases. And the reason we wanted to do that is because we know that mitochondrial DNA mutations account for the vast majority of primary mitochondrial diseases in adult patients. We know that mitochondrial DNA reads are generated during exome sequencing, but they're not routinely analyzed as part of standard bioinformatic pipelines, certainly not in the UK anyway. And we did that in a large number of patients that have been accumulated at the Institute of Neurology through various exome sequencing projects over the past 10 or 15 years to determine what the diagnostic uplift was when including mitochondrial DNA analysis during the iterative reanalysis of neurological disease exomes. And part of the reason we could do that is because of the Queen's Genomics Consortium, which is an institutional-wide consortium of principal investigators interested in exome and genome sequencing projects who've agreed to share their data. And we were able to use this data and harness this data as part of this particular project. And there are now over 14,000 DNA samples stored at the Institute of Neurology which have undergone whole exome sequencing. And this is really a fantastic resource for new gene discovery. The main challenges before undertaking this project we realized were going to be the heteroplasmy of these mitochondrial DNA mutations, so the fact that they exhibit polyploidy, and so we used a somatic variant caller. So these are routinely used for tumors to measure somatic mitochondrial DNA mutations and mosaic mutations, and so we applied this in the patients to look for low level of heteroplasmy. And the second challenge we realized was going to be so-called new mites, so nuclear mitochondrial DNA segments, which are essentially mitochondrial sequences of mitochondrial DNA that have been transposed into the nuclear genome over time. And so they can give you a false positive readout if they're interpreted as being a mutation within the mitochondrial DNA rather than actually being a false positive from the nuclear DNA. So we looked at 11,500 DNA samples stored at the Institute of Neurology. These were all exomes from blood. These were collected over an 8-year period and comprised probands with neurological phenotypes, but also unaffected relatives and healthy controls. And this is just a spread of the different clinical groups that we saw as part of this particular cohort. We used this pipeline to filter the variants. So we had a sample level filter, which essentially included the average, the coverage for the mitochondrial DNA, which we wanted to be more than 10x, and that reduced our number of samples from 11,500 to just over 6,000 unique pathogenic mitochondrial variants detected in 10,000 exomes. We then applied a variant level filter, so we just wanted to look at known pathogenic mitochondrial DNA mutations, so not those that have just been reported, but those that would be clearly reported as pathogenic in a diagnostic laboratory. And that reduced the number of unique pathogenic mitochondrial DNA variants that we detected to 28 across 83 different exomes. And then we applied an additional filter with the read depth being over 10x and the level of heteroplasmy being more than 10%, because we felt that a level of over 10% was definitely going to be abnormal. We all harbour fairly commonly low-level mitochondrial DNA mutations. We wanted to have a fairly strict threshold of over 10%, which would always be abnormal. And so we detected 24 unique pathogenic mitochondrial DNA variants across 64 exomes from our original 11,500, and then we stratified these into 3 groups. The first was those where these mutations were clearly the cause of the patient's phenotype. And so we termed these clinically significant pathogenic mitochondrial DNA variants, which occurred in 11 exomes. We found mutations in an additional 12 exomes, which didn't clearly explain the phenotype of the patient. And so we termed these variants of undetermined clinical relevance. And finally, we had a larger group of 41 exomes in which we identified pathogenic mitochondrial DNA variants, but these were mutations that often exhibit variable clinical penetrance, even at near homoplasmic or homoplasmic levels. So often people will carry these mutations and then never manifest disease. So this is just a further breakdown of these groups. So in the first category of patients with clinically significant pathogenic mitochondrial DNA variants, we identified 7 mutations in complex 1 subunit genes. So complex 1 is the largest subunit of the respiratory chain. So it's a fairly common cause of mitochondrial DNA-related disease. And we identified 4 pathogenic variants in a subunit important for complex 5, also called MT-ATP6. This is a protein coding subunit, another protein coding subunit. And these were all found at fairly high levels of heteroplasmy, despite these being adult patients. These were high levels and had often fairly severe central nervous system phenotypes, including Lee syndrome and MELAS syndrome. And then those patients with a lower level of mutations in these genes had a less severe phenotype, often with either isolated neuropathy or ataxia or retinitis pigmentosa, etc. Looking at these exomes in more detail, we wanted to understand why mitochondrial disease hadn't been considered. Given in hindsight, you might have anticipated this could have been a disorder of mitochondria. And actually, in 5 cases, primary mitochondrial disease hadn't been suspected in these 11 cases. And I think that really just emphasizes the overlap that exists with other neurological diseases, which can sometimes make the diagnosis challenging. However, in 6 cases, primary mitochondrial disease had been included in the differential diagnosis, but not confirmed prior to performing exome sequencing. And again, the reason for that, I think, it is multiple, and I'll come back to that. But I think in part, it's because of the access people have to mitochondrial DNA sequencing, which arguably people have greater access to exome sequencing than to mitochondrial DNA sequencing, and the expertise in terms of interpreting the variants that are generated from mitochondrial DNA sequencing. And the other interesting point was some of these patients, of the 6 patients, had normal muscle pathology, which again might have been a reason that those weren't, dedicated mitochondrial DNA next generation sequencing wasn't progressed in this group of patients. The second group that we looked at or defined were those with pathogenic mitochondrial DNA variants of undetermined clinical relevance. So these were patients where the phenotypes didn't really fit with a well-recognized mitochondrial phenotype, or an alternative diagnosis had been confirmed. So for instance, in 3 of these patients, they had a pathological diagnosis of multi-system atrophy, and in 1 patient, they had a confirmed genetic diagnosis of VAMP2 mutation. In fact, this was the only confirmed genetic diagnosis in the entire cohort in this particular patient, who had a neurodevelopmental disorder with hypotonia and autistic features and a hyperkinetic movement disorder. One interesting patient I wanted to highlight was a patient who had a clinical and pathological diagnosis of inclusion body myositis, a common acquired muscle disorder in the over 50s. And this particular patient had 98%, and in fact it was 100% when we looked at this using dedicated mitochondrial DNA next generation sequencing. So 100% level of the mutation in blood, which has not previously been reported. And this patient didn't have a phenotype compatible with this particular mutation, which causes, at the very high levels, MELAS syndrome. And so this is a patient that we're looking at, we're going to look at in more detail to try and understand what the compensatory mechanisms might be that means this individual isn't suffering from a mitochondrial disease. And then the final group was this large group of patients with known pathogenic mitochondrial DNA variants with very variable clinical penetrance despite very high mutant loads. So these are mutations that have been associated with optic neuropathy, with deafness or aminoglycoside-induced deafness, and cardiomyopathy. In most of the patients, they had almost 100% mutant levels, but none of them had any symptoms that would suggest that these were manifesting, apart from one patient who had deafness, but we felt that the deafness was actually more likely caused by an alternative problem. So just in terms of the discussion of this study, we reanalyzed 11,500 exomes and identified 24 unique pathogenic variants in the mitochondrial genome across 64 exomes. Of these, we felt 11 were clinically significant, so a very modest number, but given the fact that it's fairly easy to apply this this technology to routine bioinformatic pipelines, and there's no extra cost to it, we think it's worthwhile including routinely just to make sure none of these cases are missed. In 5 subjects, primary mitochondrial hadn't been considered, and again, I think that relates to the significant phenotypic overlap that often exists in other neurological diseases. And in the 6 in which mitochondrial disease was considered, the diagnosis wasn't pursued further beyond the clinical phenotyping prior to whole exome sequencing. And I think that just highlights a number of other challenges with mitochondrial diseases. So the overlap, the fact that muscle pathology can be normal, and that shouldn't really put you off the diagnosis, particularly in children. Access to mitochondrial DNA, dedicated mitochondrial DNA next generation sequencing, and particularly the clinical scientists with the expertise to interpret those variants. And the fact that not everyone realizes that mitochondrial DNA analysis is not routinely included as part of exome sequencing bioinformatic pipelines. The other groups of patients that were identified are arguably more challenging because although we didn't feel that those particular mutations were contributing significantly to the current phenotype that they had, there's still a possibility that they may develop symptoms later in life related to these mutations, that subclinical manifestations such as cardiomyopathy could be present, and these mutations have reproductive implications. So even if these patients have fairly modest levels, which aren't causing symptoms to manifest, they can transmit those to offspring who may end up with much higher levels and much more severe disease. And there are reproductive options and choices available to women with mitochondrial DNA mutations to prevent the transmission. And so these ethical considerations, we've explored these ethical considerations recently in a commentary, and really touching on the 2 major aspects which we think are a challenge and need a wider debate. And that's the fact that many of these large-scale sequencing technologies that we're now using, both exome and genome sequencing, are potentially going to diagnose people with mitochondrial disease much earlier in the natural course of their disease and before an extended phenotype is manifesting. So even if you're looking for a mitochondrial DNA mutation, it's not just a diagnostic test, it's potentially a predictive test with major reproductive implications. And that you might also detect these mutations completely coincidentally when they have nothing to do with the phenotype. And in both these situations, we feel that discussion around the implications of mitochondrial DNA mutations needs to be had with the patient so that they're aware during the counselling process of what the potential implications are of a positive genetic mitochondrial DNA mutation. There are limitations to the study that we undertook. So unlike whole genome sequencing, exome sequencing actually achieves a fairly low coverage of extracted mitochondrial DNA reads, and in particularly in comparison to those using diagnostic next generation sequencing of mitochondrial DNA, which is dedicated deep sequencing. And so you can miss low-level heteroplasmic mitochondrial DNA variants, particularly in blood. And so performing a muscle biopsy and undertaking dedicated mitochondrial DNA next generation sequencing is really the gold standard. And also the fact that spurious identification of variants within these nuclear mitochondrial DNA segments can occur, although given the fact that we simultaneously aligned the reads to both the nuclear and the mitochondrial genomes as part of this approach, and the fact that we confirmed all the mitochondrial DNA mutations using long-range PCR to enrich the mitochondrial DNA, followed by next-generation sequencing of mitochondrial DNA, the risk of false positive finding in this setting is extremely low. So conclusions to the study were that mitochondrial DNA reads are generated during standard clinical whole exome sequencing, but not routinely analyzed. We feel that analyzing these data is worthwhile, even if the diagnostic uplift is modest. And therefore, we feel that mitochondrial DNA analysis should be incorporated into the evaluation of neurological disease exomes in terms of bioinformatic pipelines. But with the important caveat that if you suspect a primary mitochondrial disease and this test was negative, you still need to go back, reevaluate the patient, consider a muscle biopsy, and repeat the mitochondrial DNA sequencing from mitochondrial DNA extracted from muscle tissue. So I think I'll stop there. I just want to thank all the people who are working across different highly specialized services in the UK, providing care for patients with mitochondrial diseases based in London, Oxford, and Newcastle, all our funders, and also all the members of the team who are very much involved in doing all the work for these projects. And Jana Vandrakova is our senior bioinformatician involved in both of these studies. Olivia Poole and Chiara Pisamiglio were involved in looking at the mitochondrial DNA from the exome sequencing data project, and Dr. Will Macken and Dr. Caroline McKintrick are involved in looking at the 100,000 Genomes project and the patients with mitochondrial disease that have been recruited to that. I'll stop there, and I'd just like to say thank you, and anyone who wants to contact me after the talk for any reason, I'd be very happy. Thank you.
Thank you so much, Dr. Pitceathly. It was really wonderful talk. I think now we can open up the floor for discussion, question and answers. So if you have any questions, please feel free to raise your hand and unmute yourself, and you can also write your questions on the chat. And while we are waiting questions from the floor, I'll ask one. It's really interesting that you have a great uplift by reanalysis of the data. How about, I was wondering, utilization of other cell types or other like tissue types? Like sometimes, I mean, having muscle biopsy is very helpful, but there's tendency to not to do muscle biopsy more and more, especially in the pediatric population. But sometimes for research projects, we do have fibroblast samples derived from skin biopsies. Do you have any thoughts about the diagnostic yield from skin samples?
So, I mean, as you see in this particular project, we looked in blood, and we do, I mean, as you say, you know, we tend to avoid muscle biopsies these days. So we do often start with fairly extensive genetic testing, even of the mitochondrial genome from blood. And then in our diagnostic service, we find actually urine very helpful. So we extract mitochondrial DNA from urinary epithelial cells, and we actually find that that correlates more closely with post-mitotic tissue than blood does. And so we frequently look at that. The major challenge with that is often because of the number of cells that you can often gather from urine. It's often prone to not working. You can't extract the DNA, but if you can, it's very useful. Fibroblasts we don't routinely look at in a diagnostic laboratory, although we look at in research studies very, very often. And the reason for that is there is a shift often in the level of the mitochondrial DNA through different passages of fibroblast cultures. And so you often see a shift in the level of the mitochondrial DNA. And so it's difficult to interpret when you receive a level what it means, and if it's negative, whether it's truly negative. And the shift can actually go in both directions, or often it does actually drift down, but it can actually go in both directions. So we don't routinely look at fibroblasts as part of our diagnostic service. We really look at blood, urine, and muscle. We will look at saliva as well, but we do look at fibroblasts often in the research setting.
Great, thank you. Okay, are there any questions from the floor? Maybe I'll ask another one. So it was great that you see this difference, but going forward, how do you, what are your recommendations? Like, is it going to be more clinician driven when they say, okay, please check the mitochondrial DNA as well? Or can you implement it as a routine in the practice, in the daily operations of the diagnostic lab?
I think at the moment, and this is something that is being wrangled as part of the 100,000 Genomes project. If you can, which at the moment is not routinely reporting mitochondrial DNA variants, because if someone isn't considered to have a mitochondrial disease, but you detect a pathogenic mitochondrial DNA variant, as I've alluded to, it raises all sorts of ethical implications, because potentially it has all sorts of implications that you can't predict, and the patient hasn't really been counselled properly during the consent process to having a genetic test and having those looked at. So I certainly wouldn't recommend at this stage routinely looking at mitochondrial DNA unless there was a strong clinical suspicion of a mitochondrial disease. I think where it's useful is in existing exome data sets where mitochondrial DNA hasn't been sequenced, but mitochondrial disease was a consideration and the patient hasn't maybe had a muscle biopsy, for looking at that and applying it in that setting. But I think it's important that mitochondrial disease was a consideration at the outset, just to avoid these ethical implications in terms of feeding back that result to the patient.
It's very hard, especially with low penetrance mutations.
Yeah, it's very difficult to predict, and that makes it very difficult when talking to the patient about the predictive implications of the test. There's no really evidence base for that, and there's no evidence base for much of the screening that we do for mitochondrial DNA mutations. So it's very challenging.
Okay, are there any questions? There are no new questions. We'll thank Dr. Robert Pitceathly for this wonderful discussion and his insight in mitochondrial disease diagnosis. Thank you so much. And we look forward to hearing more about your research in the upcoming years. Thank you.
Thank you. Thanks very much.
Thank you so much.